Yes, Dr. Lavkush, the presentation is visible.
Yes, sir. It is visible, sir.
Okay, okay. Thank you very much. So I hope it will be uh, of some interest to people who are in biological sciences, because it is little bit technical kind of presentation, and I will try to make it as simple as possible so that everybody can get it, because it is today it is for say general public as well, that everybody is looking for quality products. So if we are looking for quality products, then most of us are information most of us are aware about the quality standards. So good laboratory practices is a concept of testing drugs and pharmaceuticals so that we can get good quality products and which are say pharmacologically active. So how the concept started? So we say that it is a quality system. It is a quality system concerned with the organizational process and the condition under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived and reported. So you can see lot of words in the definition itself, and the one prime word is that it is a quality system where non-clinical health and environmental studies performed, because every drug or pharmaceutical which is coming for human or veterinary application, it must be screened for biological activity and safety, and if it is not screened properly, then we may end up with getting an in the pipeline. It may adversely affect our own health, or it may adversely affect health of our our pet animals or cattle or all that. So these are non-clinical. First word is non-clinical, means these studies are not hospitals. Hospitals idea of called like regulations are there. These GLP are only for laboratory exercises. So the studies which are conducted in laboratory are under GLP, and these are safety studies. These are studies means these are experiments, and so these experiments are non-human. These are largely on animals or other say in vitro. So these are for non-human experiments. So how GLP workflow works, that generally the pharmaceutical agency they ship drugs for analysis to laboratories, external or we say third party laboratories. These laboratories must have GLP certification. Like if our innovation center we want to do something, then we would have a GLP certification. So the data, and we will be a third party agency, because a drug is made by somebody else and we have done the analysis and then we have submitted data to the company and the government as well. So the company will give us a drug for analysis, we will analyze the drug and submit the data to the government and to the company. Now based on the assessment of the data, the hazard assessment will be made, and then the compound, if it is found safe, then it can be processed for other kind of biological activities or pharmacological activity, and then it can go in the like trade pipeline of manufacturing and all that. So why it was required? Actually in early 70s US FDA, it came to know that some of the laboratories they are performing experiments poorly. That means that either they are not recording the experiments properly, or their planning of the experiment is non proper, and we are not getting adequate data for assessment about the biological activity and safety of the drug molecule. And they also found that a lot of fraudulent activities are going on. So labs which are not equipped adequately, they are doing some experiments and then they are reporting something else. So results are something else and reports are something else. So these kind of fraudulent activities are going on. So to check these activities, the concept of GLP came. So was initiated in US also, but first GLP regulations came in New Zealand, and in US it was because US FDA is one of the best regulating agency in the world which regulates so many products. So they came to know about a case from one laboratory that is called Industrial Biosafety Biotest Lab, which is doing safety analysis of drugs and pharmaceuticals. So in one case which led to the formation of GLP rules, it was the Industrial Biosafety at the lab IBT. It was running some tests for Procter and Gamble, and these molecules, they were for cosmetic use. So they have tested these molecules for safety on rodent models, and in mice they tested these products, and it was discovered that these products were not safe for animals. But later on, I covered up the results and claimed that the products were good for human. So later on, some toxicity reports were came in during pharmacovigilance practice, and then it was like US FDA. It took action against this particular lab, and the GLP norms were formulated. GLP was first introduced in New Zealand, and in 1979 US FDA came with the GLP legislation, and then they came up with regulation which became effective in June 1979. So the purpose of GLP is that to certify that whether every step of the analysis is valid or not. Means suppose I am running a spectrophotometer and I am getting X concentration of a drug. Now whether the concentration of the drug is the same or not, or whether the test what I am running, whether the method what I am using, whether the method is valid or not. So we should have the validity of the method. Then to assure the quality and integrity of the data, submit to US FDA or any other agency in support of the safety of the regulated product. So we say that this product is safe for animal or human consumption. So FDA has to has ruled that as per GLP we have to be assured of the quality and integrity of the data. Integrity of the data means like whatever results are there, those results are reported in real. There is no masking of the results. And the third is emphasis on data recording and sample or specimen retention. So whatever results I got today, I must record that data and retain the sample or specimen. Like suppose I have done some analysis using a particular product, then that product should be retained for some time. आप लोग शायद जानते होंगे कि जैसे जो भी हम मेडिसिन खरीदते हैं तो उस मेडिसिन पे एक्सपायरी डेट है. अब उस एक्सपायरी डेट जिस कंपनी ने उस मेडिसिन को बनाया है उसकी एक्सपायरी डेट तक ड्रग को फॉर्मुलेशन को अपने पास करना होता है. कभी भी में उस ड्रग के कारण कोई भी अगर एडवर्स इफेक्ट आता है तो कंपनी को अपने उनके पास जो ड्रग रिटेन की गई है, जो सैंपल रिटेन किया गया है, उस रिटेन्ड सैंपल से उसकी सेफ्टी को प्रूव करना पड़ेगा. In case if the company is not able to prove that safety, then company will be in a big trouble. So it is the practice of GLP that every data, recorded data should be stored and the sample should be retained with which we have carried out that data. So that sample should be retained throughout the life of the sample, whatever lifetime we have given to the sample, 2 year, 3 year, 4 year. So for that lifetime of the sample should be retained. In 1981, another organization named OECD was framed, that is Organization of Economic Cooperation and Development. So it produced GLP principles that are international standards of OECD, and India is a member of OECD. So all OECD members, they have to comply with the GLP regulations. Means whatever regulation is made by say US FDA and it is passed by OECD, then it has to be followed all the member countries. The advantage is that if suppose one product is analyzed in India using GLP, India member of OECD country, then any Indian company can market product in Europe or in countries which are member of OECD. So all member OECD country, they will deem that done by a OECD country that must have been done up to the standard operating procedures or the using the GLP practices. So India became a full member of OECD. In India, I have talked about now that National GLP Compliance Monitoring Authority was established in 2002. So before 2002 it was not there. So now we are member of OECD country. So whatever analysis we are doing in our laboratories, if we have to do it for international marketing, or we have to present it at some very good platform in India as well, then it must be completed as per GLP principles. Then objective of GLP is, the first objective is true reflection. True reflection means GLP makes sure that the data submitted are true and true reflection of the result that are being obtained during the study. So whatever we got, we have to report. It's very clear. Whether the reports are negative, whether the reports are positive, the report must be true reflection of our study. Then ensure honesty. GLP also make sure that not to indulge in any kind of fraud activity by the lab. So we should not and we cannot mask interference. So if we have worked as per the GLP principles, it promotes international acceptance of the test and it helps us in saving a lot of money. Because if I have a drug in India and if I want to market it in say Germany, and if they say okay, you have to do the test and again pay for the test, then definitely I have to invest a lot. But if India being a member of OECD country, Germany being a member of OECD country, and we have done everything as per GLP and we claim that we have done everything as per GLP, then the results will be accepted in other country as well and it can save us a lot of money. Then principle of GLP apply on all non-clinical health and environmental safety studies required by regulations for the purpose of registering and licensing. So these includes drugs and pharmaceuticals, pesticides, food and feed additives, cosmetic products, veterinary drug products and similar products, and industrial chemicals. For all these category of chemicals we can do GLP. So these are 9 principles of GLP, that is test facility organization and personnel, quality assurance program, facilities for doing the tests, apparatus, material and reagents, test systems, test and reference items, performance of the study, reporting of the study result, and storage and retention of the records and materials. So I think I'll not go in detail of these 9 principles, although like I have made the presentation like that I have given detail of each and every principle. One thing that is very strictly that has been enforced in GLP is that we should have SOP for everything, standard operating procedure, and we should not deviate from SOP. So we have to write what we do. It should be written in a chronological order, and every step should be explained that how it is working. So SOP for every equipment, SOP for every experiment has to be written. So these are like different principles, 9 principles are there. So I'll just skip that part. So I'll just come to this slide. So a company is making a product, so manufactured by any drug and pharmaceutical company. It is given to the testing laboratories which are having GLP certification. So GLP laboratories, they test those products on the 9 principles, and then the data is either like it is largely for safety, either it can be toxic or it can be safe. If the drug is toxic, then further like say manufacturing or the further say we can say going in pipeline for drug development of the drug is stopped. possibly people who are in like from scientific background in this group, they understand that we have different kind of clinical trial stage. So preclinical is done on animals, and if any molecule is passing preclinical stage, then only it can go on the clinical stage. So in preclinical stage, the drugs are tested on animals, and if you find these are safe for human, for animals, it can go for human consumption. Then only it is it goes for clinical trial. But otherwise, it dies here and only at this stage. So if it is found safe, then it is going for further analysis, and then finally goes to market or international trade. So GLP in India, became member of GLP in 2002, and we got the National GLP Compliance Monitoring Authority under Department of Science and Technology, Government of India, and we have a large number of trained experts as GLP inspectors in India. So if suppose I file for GLP of certification for my facility, then it will be inspected, and then I may or may not be issued a license. So the scope of GLP application, that is NGCMA, National GLP Compliance Monitoring Authority of India, physical and chemical testing, toxicity studies, mutagenicity studies, environmental toxicity studies, and aquatic and terrestrial animals. Then studies on behavior in water, soil, air, bioaccumulation, effect on natural ecosystem, and analytical and chemical testing. So these are major areas where NGCMA is having the regulatory role. The certification process is that suppose anybody who want to, any laboratory who want a GLP certification, we have to apply to the NGCMA in a prescribed performa. Then there will be pre-inspection, then there will be final inspection, report submission. Finally, we get a certificate which will be valid for 3 years. So after 3 years, again we have to go for recertification. So this is the National GLP Compliance Monitoring Authority, located at DST New Delhi. The regulation approved as standard by OECD to avoid and the fraud activities of the testing laboratory for drug and pharmaceuticals, pesticides, food additives, dyes to save the human and environmental health, and also erect good international trade and establish good relationship among the member countries. So I found, I thought I can show it to you that the manager of the company is always thinking about zero defect, but the plate itself or the signboard itself is having defects. So we have to think zero defect, and we have to ensure zero defect. So we can only get GLP when either when we do what we write, or we write what we do. So both the things are correct. Either you do what is written in the SOP, or if there is any change in SOP, then you simply write the new SOP and you write what you have done. So thank you very much, and I hope it was not very boring for all of you. Maybe for some of you who are not from science background, but I thought I try to make it as simple as possible. So thank you very much.
Thank you so much, sir. Thank you. Definitely it was very informative, and काफी कुछ सीखने को मिला इसमें, गुड लेबोरेटरी प्रैक्टिसेस और एज वेल एज प्रोजेक्ट राइटिंग पर भी. और मैं फिर से अभी पार्टिसिपेंट से अनुरोध करूंगा, अगर किसी के कुछ क्वेश्चंस हैं, दे वांट टू आस्क समथिंग, प्लीज वो पूछ सकते हैं. हेलो, if anyone wants to ask something on GLP or project writing or project handling, they can ask, please. Okay, so I think
okay, Dr. Lokush, there are no questions. I think it was very clear to all the participants.
Yes, sir. It is,
and all my fellow colleagues.
Yeah.
So thank you, everyone. Thank you for your patient hearing, and I hope it was of some use to you. Thank you very much.
Thank you so much, sir, for giving your valuable time and delivering such an informative lecture on this topic. And thanks to all participants for joining this session. And now we are closing the session here.